بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله. أنا اسمي الدكتورة سامية درويش. Today we're going to speak about the etiology of periodontal disease. We're going to talk about the dental plaque biofilm. The oral cavity contains about 600 species of microorganisms, and it is divided into it is present into 5 major ecosystems or niches. The niches are hard surfaces, periodontal or peri-implant pocket, the epithelium, the dorsum of the tongue, and the tonsils. If you look on the right side of the slide, you will find the dentogingival junction, which is composed of 1 junctional epithelium, 2, on the number 2, sulcular epithelium, and the pink 3 oral epithelium, and 4 epithelial rests of molasses. The junctional epithelium functions are attachment to the tooth, it acts as a barrier, and has a rapid turnover. That means the epithelium changes and renews rapidly. And it has an antimicrobial defense, and it produces gingival or crevicular fluid into the sulcus. At the bottom, you have a diagram which represents the gingival sulcus, the free marginal gingiva, and the marginal groove. Between the marginal groove and the mucogingival junction, we have the attached gingiva, and apical to the mucogingival junction, we have the alveolar mucosa. Now, periodontal disease is primarily caused by bacteria. This is the major etiologic factor. However, the host also plays a role in the progress and the severity of periodontal disease. But without bacteria, you will not have periodontal disease. And depending upon the host response, we will determine the severity of periodontal disease. All right, the etiology of periodontal disease is multifactorial. This diagram represents the multiple factors which cause periodontal disease. The primary etiological factor, as I said, is plaque and the pathogenic microorganisms present in plaque, represented by antigens, lipopolysaccharides, and virulence. And to combat the plaque, which is a major etiological factor in periodontal disease, we have a host response. We have the inflammatory immune response, which is represented by polymorphonucleoleucocytes, antibodies, and complement as a first line of defense, and the macrophage and lymphocyte axis, which also represents the second line of defense. The polymorphonucleoleucocytes are phagocytes, which are going to phagocytose the bacteria, and also the antibodies are going to deal with the bacteria. The inflammatory immune response will result in the liberation of cytokines, prostanoids, matrix metalloproteinases, which are going to produce connective tissue destruction and bone destruction. These 2 factors, the plaque and the host, are going to give us the clinical symptoms of disease. So if the plaque is aggressive or the connective tissue is hyper-responsive, these 2 factors are a balance which is going to give us the nature of the clinical symptoms of the disease. We have to also consider genetic factors which play a role. Example, trisomy 21, like Mongolism, and non-genetic factors like smoking and diabetes mellitus. To demonstrate the effect of plaque accumulation on gingivitis, the Danish Professor Harold Lowe in very early 1965 told his students who had clean teeth and no gingivitis to stop brushing their teeth and then reinstate brushing. Now this diagram represents on the x-axis the days during which the students did not and then reinstated their brushing techniques. And on the y-axis, we have the gingival index, the severity of gingivitis. The blue color represents gram-positive organisms, and the red color represents gram-negative organisms. And of course, on the bottom, we have spirochetes. All right. Now, at the beginning, when they had brushed their teeth and no gingivitis, we can note that the gram-positive organisms represented by the blue color was only present only to a little extent. Then as these students stopped brushing their teeth, the gram-negative organisms in red start to accumulate and the gingival index started to increase. That means the gingivitis started to show. With the progress of no brushing, then more gram-negative organisms and also gram-positive organisms and spirochetes accumulated, and gingivitis reached its peak. Then after the day 20, he started to ask them to rebrush their teeth again. And what happened is that when they cleaned their teeth from dental plaque, gram-positive and gram-negative organisms started to decrease, and the gingivitis started also to disappear. So this says there is a cause and effect. With the accumulation of plaque, you have gingivitis. With no accumulation of plaque, the gingivitis disappears. This is a classical research to demonstrate the relationship of plaque and gingivitis. All right, dental plaque is a structured, resilient, yellowish-grayish substance that adheres tenaciously, very tenaciously, closely to the intraoral hard surfaces, including removable and fixed restorations and implants. Plaque is essentially composed of bacteria in a matrix of salivary glycoproteins and extracellular polysaccharides. This matrix makes it impossible to remove the plaque by rinsing or use of sprays. So, dental plaque adheres not only to hard tooth surfaces, but also adheres to retentive factors, like, for example, in an overhang, or can reside inside pockets. Right? This is retentive factors in the oral cavity. Also, it can also adhere onto calcified dental plaque, which is termed calculus. On the surface of calculus, there is a metabolic active dental plaque. So it is only the dental plaque that causes disease. Dental plaque is calcified does not, but it is the plaque that is laid on top of it. And also irregular surfaces are good intraoral niches in the oral cavity for the accumulation of plaque. All right, we have to understand that the oral cavity harbors more than 600 strains of microorganisms, and the dental plaque, if you take like a gram of dental plaque, you find that it harbors about 10 to the power of 11 microorganisms in wet weight. So we have to differentiate between materia alba and calculus. Materia alba are soft accumulations of bacteria and tissue cells that lack the organized structure of dental plaque, and it is easily displaced with a water spray. Calculus, however, is a hard deposit. It's formed by mineralization of dental plaque and is covered by a layer of unmineralized plaque. Dental plaque is classified in supragingival and subgingival. The supragingival plaque is found at or coronal to the gingival margin, When in direct contact with the gingival margin, it is referred to as marginal plaque. The subgingival plaque, however, is found apical to the gingival margin between the tooth and the gingival pocket epithelium. Now, this is a photomicrograph of a 1-week-old plaque, and you can see that it is a dense mass of plaque. As I told you, 1 wet gram of dental plaque will be equivalent to 10 to the power of 11 bacteria. This is a 3-week-old plaque in which you see in the outer surface something that looks like filaments with cocci on top of it, and this is what we will talk about as corn on the cobs in a little while. The intercellular matrix, we knew that dental plaque is basically almost 90% composition is of bacteria, but it is present in an intercellular matrix, plaque matrix, which is composed of organic material and inorganic material. The organic material is derived from saliva, clavicular fluid, and bacterial products. These include polysaccharides produced by bacteria, for example, dextrans, glucans, very sticky material. That is why it cannot be easily removed by mouth spray. It must be removed by mechanical means, and glycoproteins from the saliva. B, the inorganic material, which is essentially calcium, phosphorus, which predominate, and trace amounts of minerals, example sodium, potassium, and fluorides. All right, I wanted to show you a very interesting phenomenon in supragingival plaque. In long-standing supragingival plaque near the gingiva, you can find perhaps, this is an electron microscope, an electron microscopic picture or figure with a filament on top, in the middle, filament in the middle, fusiforms, and on top of it is cocci, perhaps gram-positive Streptococcus sanguis, or maybe gram-negative gram-negative P. gingivalis, which gives you the appearance of corn on the cob. Corn on the cob in Arabic means Kuz il-Dora. It is a result of a cell-to-cell recognition of genetically distinct partners that adhere to each other. On the other end, we have Ichinella corrodens, which is present on a blood agar plate. I just wanted to show you how it looks on a morphological basis on the plate culture. Now, this is another picture of a dental plaque, which is, as you see, is present in a gelatinous interbacterial matrix, very sticky and very tenacious, cannot be removed by mouth rinse, only by mechanical means. It obtains, the colonies obtains its nutrients from the interbacterial matrix, but also can obtain its matrix through channels which communicate with the microcolonies together. It is firmly attached to the dental pellicle. The reason why systemic antibiotics should be adjunctive to conventional periodontal treatment and not as a monotherapy, it is because of the following reasons. The resistance of bacteria arranged in biofilms to antibiotics in biofilms. Also, an antibiotic strength of 1,000 to 1,500 times greater than the systemic therapeutic dose may be required to be effective on dental plaque bacteria because it is bound by this interbacterial matrix. To have access to periodontal pathogens, it is important to physically disrupt the biofilm. Now, this is a diagrammatic representation of a subgingival plaque. Now, we have the so-called tooth-attached plaque and the epithelial associated plaque. The tooth-attached plaque is primarily made of gram-positive organisms, namely Actinomyces viscosus, Actinomyces nislandi, and also Streptococcus sanguinus. This part, the tooth associated, is concerned or is related to the formation of subgingival calculus when it calcifies. Also, we know that Actinomyces viscosus is responsible for the development of root caries. However, the tooth-associated or epithelial-associated plaque is basically gram-negative organisms, and in the bacteria within the pocket or the gingival, in this condition it is the pocket, are basically gram-negative organisms, like for instance P. gingivalis, treponema also can stay there because they cannot attach. They must have a place to stay in. They cannot attach to tooth surfaces. Also, the gram-negative organisms are there because of the ecological determinants of subgingival plaque, where there is a reduced amount of oxygen, so anaerobes can grow there, and also mechanical influences that can entrap the motile organisms like Treponema denticola, and also because of nutritional facilities from the gingival fluid, which constantly flows into the gingival sulcus or pocket. And also we have bacteria can even progress to the bone, the tissues, and up to the bone. The unique situation or the unique environment of a subgingival plaque is that oxygen is diminished, so anaerobes can grow and multiply. So, in essence, ecological factors in periodontal pockets favor the growth of periodontal pathogens because of the following reasons. Increased redox potential, which means that oxygen is diminished and anaerobes start to be able to grow and multiply in this field. Increased gingival fluid due to inflammation, resulting in increased pH and nutrients suitable for bacteria to grow there. Also, there is mechanical entrapment of motile organisms, example spirochetes, and the anaerobic environment, which encourages anaerobes to grow and multiply in the subgingival area. The stages of dental plaque formation. First stage is formation of dental pellicle. The second, initial adhesion and attachment to bacteria. The third, colonization and plaque maturation. Now, the first stage, the formation of pellicle, within nanoseconds after polishing the teeth, a thin saliva-derived layer called the acquired pellicle covers the tooth surface. It basically consists of glycoproteins, proline-rich proteins, phosphoproteins, histidine-rich proteins, etc. And this can function as adhesion sites for bacteria or receptors for bacteria. Now, bacteria is part of the very early deposits within seconds after prophylaxis. The mechanism involved in enamel pellicle formation include electrostatic, van der Waals, and hydrophobic forces. The second point is initial adhesion and attachment to bacteria. Phase 1, transport to the surface by either random contact and chemotactic activity. Phase 2, initial adhesion, and this is reversible by van der Waals, electrostatic and repulsive forces. Now, phase 3 is attachment. You have to bear in mind that any bacteria that is unable to attach will not colonize dental pellicle. It should be able to attach. The number 2, since dental pellicle there is oxygen all around it, so the organism should be able to live in oxygen. So it has to be facultative at least. So without these two factors, it will not be able to colonize the clean, glycoproteinaceous dental pellicle. So it has been shown that organisms like aviscosis, Actinomyces viscosus, has fimbria yani أصابع, and to these fimbria it has a adhesive material which can adhere to receptors of the pellicle and thereby attach to it. Other organisms also, which are facultative, can also colonize and form the first layer of, or the primary colonizers and first layer of dental plaque, namely Streptococcus sanguinis, not sanguis, now new, aviscosis. They bind to the proline-rich protein and other receptors of pellicle. This is a diagrammatic representation of plaque formation. At the bottom you have the dental pellicle, which is a non-shedding surface, the tooth surface, which is the non-shedding surface over which is acquired pellicle. Now the acquired pellicle has the receptors upon which adhesins, as I said before, stick to it. And we said also that the only organisms that colonize the first layer of plaque should be able to live in oxygen, maybe facultative, and able to attach. So on the receptors of acquired pellicle, adhesins from primary colonizers will start to accumulate. Now, the first colonizers will also provide receptors for more adhesins to accumulate, and so forth and so on, until the oxygen in the area becomes low, and then we get anaerobes to start to grow and multiply. The Fusobacterium nucleatum, which is present as this long cigar filament organism, is an attachment or a connection between primary colonizers, which are able to live in an oxygen environment, and secondary colonizers that are totally anaerobic, and they represent secondary colonizers. All right, not only is this important, but what is important too is that we have what we call nutritional interdependencies between bacteria. For example, bacteria like Porphyromonas gingivalis and Prevotella intermedia, they depend for their nutritional requirement hemin, all right, which they can get from the host, and also they can get from other bacteria of protoheme, for example, that can be a nutritional value for Porphyromonas gingivalis. Also, something like Treponema denticola, requires as a nutritional requirement alpha-2 globulin, which they can get from the host, or can also require isobutyrate, which Porphyromonas gingivalis can provide for the treponema, etc. Minidione is also very important for Porphyromonas gingivalis and is obtained from certain bacteria like Veillonella, and can be obtained also from the tissues, and vice versa. This is what we call nutritional interdependencies. All right, the third point is colonization and plaque maturation. So, increasing binding sites to primary and secondary colonizers, as I have shown you in the previous figure, and an ecological shift to an oxygen-deprived environment, and nutritional interdependencies. We said there are binding sites between primary and second colonizers. Accumulation will result in oxygen use, and it becomes oxygen deprived, and it will allow non-anaerobic organisms to grow and multiply. Then we get the nutritional interdependencies, as I've shown you before. Now, This is a scanning electron microscope of dental plaque, showing what I showed you before is the corn on the cob with the central filament to which is attached a gram-positive cocci. Complexes of periodontal organisms, when analyzed, and an analysis of greater than 13,000 species in 40 subgingival bacteria using DNA hybridization shows that it is presented in microbial complexes. They gave it different colors. Now, they found out that the organisms which are present in the orange complex and the green complex and the red complex are pathogenic as compared to the yellow and blue and purple complexes, which are not pathogenic. The red complex, which is formed by P. gingivalis, Porphyromonas gingivalis, Tannerella forsythia, Treponema denticola. This also is associated with bleeding on probing. Bleeding on probing is a sign of periodontal disease, and therefore they were considered as the pathogenic organisms, in addition to also members of this group, the yellow group, and members of the green group, especially Aggregatibacter Actinobacillus commitans. The high density of bacteria in biofilm facilitates the exchange of genetic information among cells of the same species and across species and even genera by means of three ways: transformation, conjugation, and transduction. Transformation, when pieces of DNA from the environment into the bacterial chromosome travel to another one, and conjugation by sex pilus, and transduction by a bacteriophage. All right, this diagram represents the gene transfer for virulence, as has been described. On your left side, you have the red bacteria containing the virulent factor. The virulent factor is present in a circular DNA molecule. So the DNA carrying the virulence can be transformed from the virulent to the non-virulent by transformation, as in case of the left-hand side A diagram, and this will render the non-virulent into a virulent organism. The circular DNA molecules here, the plasmids here on the second slide, which is virulent with the virulence factor, a DNA carrying the virulence factor, can be also transferred via a sex pilus into the non-virulent organism, rendering by conjugation this time, rendering it into a virulent organism. The third way of transferring the DNA molecules containing the virulence factor is by the bacteriophage, which carries the DNA carrying with the virulence factor into a non-virulent organism, transforming it into a virulent organism by transduction. Now we know that the subject variation in plaque formation can be explained by factors like diet. If you have a high carbohydrate diet, you're liable to get more plaque formation. And chewing fibrous food renders you less liable to get a plaque formation. Smoking, of course, makes you more liable to accumulate plaque. And also tongue and palate brushing. Sometimes people don't brush their tongues, and this will result in more plaque formation on the tooth surface. This is a culture of a Porphyromonas gingivalis and also Bacteroides intermedius, and these are black pigmented colonies on selective media with minadione and hemin, which is a nutritional requirement for these types of bacteria, and gives you black colonies for identification and from other colonies. And this is it's not Actinobacillus now, Aggregatibacter Actinobacillus Actinomycetum comitans, which is a star-shaped colony, and this is a selective medium which inhibits all other bacteria, but itself it's made of bacitracin, vancomycin, and this is the colony and how it looks, a star-shaped colony. Now, an important thing that we would like to discuss is, now, sometimes plaque formation is not very much, but yet we have a lot of destruction, a lot of bone destruction, as you can see here in the radiographs. This is a 22 years old black female with family history of early tooth loss from periodontal disease. So what does this make us think? Now, we said that plaque causes periodontitis and aggressive disease, but however, here we have only minimal amount of plaque and we have aggressive disease. Bacteria associated with health and different disease entities. Gingival health. 2/3 of bacteria are gram-positive cocci, Streptococcus sanguinis, Veillonella parvula, and Capnocytophagus ochracia. 2. Gingivitis. There is increased number of filamentous rods, gram-negative bacteria, and spirochetes. In chronic periodontitis, we have gram-negative rods and anaerobes predominate in active sites. Now, if you look at the names of organisms, you find that they're all written in italics. The genus in capital and the species in small letters. For like you see here, Porphyromonas gingivalis, P capital, G in small letters in italics. So, Porphyromonas gingivalis, Prevotella intermedius, Tannerella forsythia, Aggregatibacter actinomycetum comitans, Campylobacter rectus, E. corrodens, spirochetes. I want to go back to gingival health. Now, as you see that Streptococcus sanguinis is present in health, I wanted to just say that this particular organisms elaborates hydrogen peroxide, which is lethal to AA, Aggregatibacter actinomycetum comitans. So it is present in health, and also it is antagonistic to a pathogenic organism. Now, in inactive or successfully treated sites, we get small number of the above and increased number of Streptococcus sanguinis, Veillonella parvula, and Capnocytophagus ochracia, which are present in gingival health. The predominant microbiota in different periodontal disease entities. In localized aggressive periodontitis, we have the Aggregatibacter actinomycetum comitans. In generalized aggressive periodontitis, we have Porphyromonas gingivalis, AA, Tannerella forsythia. In acute necrotizing ulcerative gingivitis, we have predominantly Porphyromonas gingivalis, Prevotella intermedius, Fusobacterium, and spirochetes. In refractory periodontitis or periodontitis that does not respond to periodontal therapy, we can get a combination of these organisms. You'll know later on. The criteria for identification of periodontal pathogens. Now, the Professor Koch he termed it now Koch's postulates. He stipulated the following for the causative agent. That means an organism that can cause disease must have the following criteria. It must be routinely isolated from diseased individuals. 2, it must be grown in pure culture in the laboratory. 3, it must produce a similar disease when inoculated into susceptible laboratory animals. And it must be recovered from lesions in a diseased laboratory animal. However, if you look at the first criterion, Koch's criterion, that means what he said about it must be routinely isolated from diseased individuals. In case of periodontal disease, this is a different matter. For example, to follow Koch's postulates, diseases like, for instance, diphtheria, if you isolate Corynebacterium diphtheriae, then it is diphtheria. If it is not there, then it is not. If you have, for instance, tuberculosis, and you isolate Mycobacterium tuberculosis, then the disease is tuberculosis. If it is not there, then it is not. Treponema pallidum, for instance, in syphilis, if it is isolated, then the patient is syphilitic. In periodontal disease, it is not the case, because organisms can be present in health, but in smaller proportions, and they inhabit a great number of surfaces, like the tongue. It can inhabit other places in the oral cavity. So it is a matter of proportion and not a plus or minus, or cultivable or non-cultivable, or detected or not detected. However, if we see what happens in the oral cavity, we can find pathogenic organisms like P. gingivalis, Porphyromonas gingivalis, and Actinobacillus and Aggregatibacter actinomycetum comitans in supragingival and subgingival plaque. It can be present in saliva, right? It can be present on the tongue, on the dorsum, on the lateral border, as you see here. Aggregatibacter is present too much on the dorsum of the tongue. It can be present in the mucosa, and it can be present in the attached gingiva. This is estimated by % positive DNA probes. What happened? Now, we cannot follow Koch's postulates, because the organisms does not necessarily be absent from disease. It is present in healthy individuals, but in small numbers in different parts of the oral cavity. This made Sokransky, my professor, to propose criteria by means of which periodontal microorganisms may be judged to be potential pathogens. So this is his view. It must be associated with disease, increases in number of organisms at disease sites. It must be eliminated or decreased in sites that demonstrate clinical resolution of the disease with treatment. 3, it must demonstrate a host response, alteration in host cellular or humoral response. It must be capable of causing disease in experimental animal models. It must demonstrate virulence factors and cause destruction of periodontal disease. And these are the data to support Aggregatibacter actinomycetum comitans and Porphyromonas gingivalis as periodontal pathogens based upon these criteria. There are different hypotheses in the etiology of periodontal disease. Do we have a specific plaque hypothesis? That means specific kinds of organisms are only responsible for the disease, or a non-specific plaque hypothesis, which indicates it is the amount of plaque that is responsible, or what we call the ecological plaque hypothesis. What actually follows the specific plaque hypothesis is we find great numbers of certain organisms, as we have seen in diseased sites, especially aggressive disease, and this supports the specific plaque hypothesis. However, what supports the non-specific plaque hypothesis is Lowe's experimental gingivitis in man, when accumulation of plaque led to gingivitis and disappearance of plaque, gingivitis disappeared. So this can be attributed to gingivitis only. However, the ecological plaque hypothesis indicates that not only the bacteria is important, it is also the ecological ecology of the oral cavity. Ecology يعني العوامل البيئية. For example, if we eliminate plaque, we can not ensure health except when we eliminate the ecological niches, places where plaque accumulate, like, for example, ill-fitting margins, irregular teeth, many other, also we can have diseases that can have low detrimental host response, like diabetes, or habits like smoking. So all these have to be rectified in order for us to, or taken into consideration in order for us to have a healthy periodontal tissue. So plaque has to be removed, also the ecology of the oral cavity and the system has to be also put into consideration when we want health. Let us see Sokransky's hypotheses or criteria, and see, not Actinobacillus now, Aggregatibacter actinomycetum comitans. It is associated with disease. When eliminated, yes, disease is removed, and it gives a host response. It does possess virulence factors, especially leukotoxin, and can produce disease in animal studies. Also, Porphyromonas gingivalis, the same thing, it is associated, and when eliminated, disease progression slows down, and it has a positive host response, and it has virulence factors and can produce disease in animal studies, similar with Prevotella intermedia and Fusobacterium nucleatum to a certain extent, and Tenorella forsythia, etc. Here we go, highly pathogenic, AA, Porphyromonas gingivalis, Tenorella forsythia, Treponema denticola, moderately pathogenic, P. intermedia, Campylobacter rectus, and Treponema species, slightly pathogenic, Streptococcus intermedius, etc. Anaerobic gram-negative pathogens include, I want you to see the name so that you are able to memorize them. Porphyromonas gingivalis, this is an obligate anaerobe. Porphyromonas intermedius, this is also an obligate anaerobe. Fusobacterium intermedius, obligate anaerobe. Tenorella forsythia, however, this Tenorella is after the name of Ann Tenor. She was my colleague with Sokransky, our teacher. It is an obligate anaerobe. Spirochetes, this is also an obligate anaerobe. Aggregatibacter actinomycetum comitans, this is a facultative anaerobe and is a capnophilic. That means it grows in the presence of carbon dioxide, or requires the carbon dioxide for its growth and multiplication. Fusobacterium nucleatum, obligate anaerobe, and Campylobacter rectus, which is also an obligate anaerobe. This is also a diagram of a guinea pig, to which is added a single organism, a pathogenic organism, and it shows that it can produce periodontal disease like bone loss in a guinea pig, mono-infected with the organism. Okay. The principles of bacterial transmission, translocation, or cross-infection. Bacteria are transmitted between humans or animals and humans. All right, but contagion is the likelihood of a microorganism to lead to disease after being transmitted from an infected to an uninfected host. Intraoral transmission of bacteria from one niche to another, or one place to another, is also called translocation or cross-infection. Streptococcus, just to show you examples, Streptococcus mutans, which is responsible for caries, as you probably know, was found to be transmitted to neighboring teeth. Lush, 1979. AA was found to be transmitted by the periodontal probe in patients with localized aggressive periodontitis only temporarily. In shallow pockets, maybe in deep pockets it would stay because it has nutrients for it, and also in partially edentulous patients, teeth may act as a reservoir for recolonization or colonization of the subgingival area around implants. Another thing, in guided tissue regeneration, membranes were contaminated in patients with remaining multiple deep pockets and pathogens as compared to patients with remaining healthy teeth. Now, this led to a researcher who indicated that we have to try something, because when we do a complete quadrant by quadrant scaling and root planing in a patient, we do one quadrant and let the patient come again after a week, and during that week he states that we can have plaque accumulation on the scaled and on the scaled quadrant. So we go to the next quadrant, and then we have also translocation again to the clean surface. So he said, why don't we try to do what he called a full mouth disinfection in 24 hours, so that we do not give a chance for bacteria to be translocated from one place to another, and we get a full clean mouth. So in this he did. The tongue should be taken into consideration too, because bacteria colonize the tongue and can be translocated from the tongue to the tooth. And also, we give also antiseptics, and then we'll show you how it is. So antiseptics that like chlorhexidine, tongue cleaner, also sprays, antiseptic sprays, etc. Okay, for the full mouth disinfection, as I said before, we had to clean all surfaces of the teeth and not by quadrant, because by quadrant the microorganisms of the non-clean quadrant will be translocated to the clean one. So Leeuwenhoek advocated the full mouth disinfection. So not only the teeth should be cleaned, but also the dorsum of the tongue, which carries a lot of bacteria and can be translocated to the clean tooth surfaces, must be brushed. On your right hand side, you can see a tongue brusher, but you also can use an ordinary toothbrush. You must brush your teeth after you have brushed your tongue, the dorsum of the tongue, after you have brushed your teeth. And there are other instruments, interdental cleaners, and they put like chlorhexidine gel in order to use it as an antiseptic while they clean a full mouth. So now we have a full mouth disinfection, and we have less liability of a translocation of bacteria to the clean surfaces. To sum up, we said that periodontal disease is multifactorial. We have the primary etiologic factor, which I said are bacteria, the anaerobic bacteria, gram-negative bacteria, which possess virulence factors, and the load bacteria, which is also equally important. And this, in addition to host immunoinflammatory responses, will determine the extent of connective tissue and bone destruction, which will then give me the clinical signs of disease and progression. Not only is this important, we also have environmental and acquired risk factors and genetic risk factors, as I have described before. The learning objectives is to enable students to understand the multifactorial etiology of periodontal disease with the bacteria as the primary initiator. It is also to understand the association of dental plaque and gingivitis as represented by the classical study of Van Loo, 1965, to differentiate between dental plaque, materia alba, and calculus, to recognize the structure and composition of dental plaque biofilm, to understand the sequential phases of dental plaque biofilm formation at the ultrastructural level via pellicle, namely pellicle, initial bacterial adhesion, attachment, colonization, and plaque maturation. Also to understand the bacterial transition from gram-positive to gram-negative as a result of physiological transition in the developing plaque, namely lowered redox potential of the plaque environment, nutritional interdependencies between different species of the developing plaque bacteria and the host. Understand the principle of bacterial transmission, translocation, and cross-infection, which led to one-stage concept introduced by Leuven et al. 1990. Understand the specific, non-specific, and ecologic plaque hypothesis in the etiology of gingival and periodontal diseases, and that periodontal disease is a mixed infection. Define the criteria for identification of periodontal pathogens, Koch's postulates, and the modified Sokransky's postulates, to recognize that the predominant microbiota in periodontal health and in different gingival and periodontal disease states is different. Details of the microbiological profile of the different gingival and periodontal disease states will be presented in more details in the respective lecture. The reference, Carranza's Clinical Periodontology, the 10th edition. Thank you for listening. Hope you understood in these conditions.